right, guys, good morning. All right. Brianna. Drew.
Here.
Bria. Here. Henry. Anna. Anna. Nick.
Here.
Abby. Hannah. Rosa. Here. Matt.
Here.
Jillian. Here. Mia. Here. Annie. Brianna. Brandon. Sadie. Jessica. Jesse. Where is everyone?
Nationals.
William. Marissa. Anna. Sophia. Aubrey. Here. Matthias. Colin.
Here.
Olivia. Here. Braden.
Here.
Braden, your advisor is Dr. Martin, and I'm your co-advisor for this semester because he's on sabbatical, so we should probably meet and talk about just next steps sometimes. Amber. Katie. Kennedy. Here. Ben.
Here.
Ava. Kendall. Here. Emily. Here. Lauren. Jennifer. Here. Isabel. Here. Corwin. All right, half the class is missing. If you did not pick up your exam, you can do so after class. So we stopped somewhere around. Do you guys have any questions on any of this stuff that we talked about? We talked about dendritic cells and macrophages being the two types of cells that are antigen presenting. They can present antigens to T and B cells. Antigens are small components that are part of bacteria that the immune cells can recognize. And so we stopped here, and I wanted to go over what inflammation is, like what's actually happening when you have an inflammatory response. When you have inflammation, it's technically a good thing. It means your body is doing its job. It's a critical immune defense mechanism. And so by basically having this inflammation, which is essentially this extravasation or diapedesis. So basically the blood cells, all of those are all being recruited to that area. And so that provides an opportunity for other immune cells to be recruited to the site of infection. And then so then that results in inflammation. The 5 signs of inflammation that you can see outside of the body are, if you touch it, it might be a little bit hot to the touch. Edema, which means swelling. You'll see redness, might be painful, just altered function. A change in structure indicates a change in the function. So here's an example. Say that you cut yourself with a splinter, and so there's like microbes on the splinter. It's not clean, and so you're introducing pathogens. There are macrophages right here that essentially will try to engulf the pathogen, and then they start to release these cytokines. We talked about what cytokines are. They're these small molecules that are produced by immune cells, also by epithelial cells. They're just produced by the body in many ways to kind of signal that there's something happening, and so they're these chemical alarm signals. The There's this vasoactive factor, basically, where you have, the redness and the swelling, and the blood starts pumping, and so that helps deliver even more phagocytes to help. They're all recruited to help, tackle this infection. And so then that helps then to destroy the pathogen and heal the site of inflammation. But part of this process is why we get inflammation. It's because of all of those immune cells being recruited, those cytokines are contributing to the inflammation as well. Any questions so far? So a fever, you guys should know that the normal temperature range in the body is about 98.6. And so this is a consequence of, some sort of metabolic activity in the body. So if you have a fever, then there's clearly something that's going on. Pyrogens cause fever. They are a group of chemically diverse substances. So you don't really need to know too much detail about what that means. But the reason why you get a fever when you have an infection is because your body is trying to reduce microbial growth. So that helps, the elevated temperatures help with that. So yeah, obviously you know that if you have a fever, that means there's some sort of infection going on, so it's important your body is responding in that way. So inflammation and fevers are similar processes. Okay, who here knows what a pattern recognition receptor is? Okay, so pattern recognition receptors are receptor sites on cells that recognize specific patterns on microbes. So basically, microbes or pathogens basically have these microbe-associated molecular patterns or pathogen-associated molecular patterns. So basically, these are some sort of tag, some patterns. So usually I think of it as like a tag on the microbe that can be recognized by the host immune cell. Now, the host immune cell recognizes it through these pattern recognition receptors. So there are different types of receptors. There's toll-like receptors, knob-like receptors. We don't need to get into that. This is an immunology class. But you guys should know the difference between what a MAMP or PAMP is. This is the microbe part. It's the actual tag or pattern on the microbe that is recognized by the immune system. So the structure that the microbe has is the MAMP or PAMP. They're used interchangeably. And then the pattern recognition receptors are these TLRs, R for receptor, that are recognizing those things on the microbe. So for instance, remember we talked about the flagella. Flagella, it helps microbes do what? Move, yeah, swim and move. So that is a MAMP or PAMP that is on some microbes. So some immune cells can recognize the flagella portion of the microbe. So here we see flagellin, so this is the protein that makes the flagella, and this is the MAMP or PAMP on the microbe, and this is the receptor on the immune cell. So as you'll see, there's other types of things that are on microbes, even in microbes, like the RNA, the DNA, that can be recognized by other receptors on the immune cell. They can be extracellular, they can be intracellular, but the key here is that you'll notice that these colors are the same as the color of the MAMP or PAMP. So one thing to know here is that these pattern recognition receptors are specific for these MAMPs or PAMPs. So it's not a one-size-fits-all. So now how does this happen, the process? So basically, now I'm going to be using these terms, MAMPs, TLRs, so I need you guys to understand, when I'm saying TLR, I'm talking about the pattern recognition receptor. Remember, R receptor, that's on the immune cell. So the MAMP, so over here there's different types of MAMPs. So different types of things on the microbe that are associated with its pathogenesis. And so those will bind to the specific receptor. And then that sends a signal. Basically, signaling pathways are induced. You have cytokines that are starting to be produced. And then if, for instance, it's something that gets into the cell, so these are toll-like receptors. They are on the outside of the cell. Nod-like receptors are for intracellular pathogens. So that's the difference there. So here we have the pathogen coming in. It has a MAMP that is recognized by the nod-like receptor, this PRR. And so that can also activate cytokines to be made. But essentially, this triggers this inflammasome. So just from the name, you guys should know that it's associated with inflammation and more cytokine production. And so that's kind of just a general idea of how this happens. But remember, pathogens can be extracellular or intracellular. And there are different types of PRRs, pattern recognition receptors, that can recognize them. So the cytokines, we've mentioned them a lot, but essentially what they are these proteins that are messengers that tell the cells what to do. They can bind to specific receptors. They can help signal, for different things to come along and be recruited. They can act locally or they can act systemically. They can have just different effects. So they're essentially those messengers that tell the cell what to do next. And once again, they're produced by many different types of cells, including immune cells. And so their production is regulated really well. And they help to either, cytokines can be promoted or made to turn the immune response up or turn it down. And so the thing that you guys should know about cytokines as well is that too much of a good thing can be bad. And so if you have out of control cytokine production, that can be fatal. We call that a cytokine storm. So you have hyperactive immune cell activity, your blood vessels start to leak, you start to get multiple organ failure, and you could die. So this is actually one of the causes of death in the COVID-19 virus. It's the cytokine storm where your body is just over-activating, hyperactive in making these cytokines that then starts to recruit all these different immune cells and you get sicker. Okay, any questions? All right, well, this section is done, but we're going to move on to pathogenesis and microbial diseases. So I'm going to reintroduce some things. So here, these microbe-associated molecular structures or pathogens. There's different types of things that can be MAMPs. Like I said, they could be the polysaccharides in the cell walls of bacteria or the teichoic acids. Those are in gram-positive cells. It could be the DNA or the RNA in yeast. It could be, parts of the cell wall. Flagellin is a protein that contributes to bacteria. So there's different things that can be recognized by the pattern recognition receptors. And so just again here, just showing you the same thing, just a different way to show it. Here we have bacteria, and they're triggering these macrophages to engulf, and then it releases cytokines, chemokines, and then that results in vasodilation and increased vascular permeability, which is essentially what is going to cause that redness, that heat, that swelling. And that helps recruit inflammatory cells to that area. They start to basically release these inflammatory mediators, and those are what cause the pain that you might feel, but they are doing something to help, heal that wound or that infection. Okay. So now we're going to get into some diseases, some pathogenesis. So we worked with Staph aureus in the lab a lot. And so the one that we work with in the lab is non-pathogenic, but Staphylococcus aureus is actually a very common infection that is found on the skin surface. And right now we can, probably culture it off of our skin and it's commensal. It doesn't cause any harm, but it can, if you have a cut or a scrape and it gets into those deeper layers, then it can cause a deep bacterial infection, and that's called cellulitis. So this is kind of what it might look like. This is actually a cause of death for a lot of people that are older and have these infections and they can't control them, because once you have the infection enter a wound, it's getting into the bloodstream, and so that can make it difficult to treat, especially in the elderly when their immune systems are already knocked down. So you should know cellulitis and the microbe that it's associated with. Remember, Staphylococcus aureus is gram-positive cocci. You guys have seen this in the lab. Now switching gears a little bit, hookworms are another type of infections, and they are helminths or parasites. They usually are introduced into the body through the skin, typically the feet. If there is some sort of fecal matter that has the hookworms, then that's how you can get it into your body, and you actually can see the worm in the foot. And those then make their way to the intestines through the bloodstream, and they use their teeth to attach to the intestinal wall, and then they suck your blood from just your blood vessels and capillaries. And then they secrete things, anticoagulants, basically things that allow the blood not to coagulate. So it keeps flowing, and so it can then, keep sucking that blood and feed on your blood. So that infection obviously results in some inflammatory response, but these hookworms need to select a new feeding site every 6 hours. So hookworms, they are another type of infection. I'm just kind of going to throw random ones at you. Yeah.
If you get a hookworm, how do you get rid of it? Is it surgically, or do they have to
I think there are some medications that you can take that would help clear it. Yeah. Yeah. So. All right. Next is Ebola. Ebola and hemorrhagic fever. Ebola is a very contagious virus that causes extensive hemorrhaging. Does anyone know what hemorrhaging means? Yeah.
Is it within your brain?
Yeah, so when you a hemorrhage means you're bleeding out essentially. And so you can have a brain hemorrhage, which means that there's blood in the brain. But you can have intestinal hemorrhaging, so that's if you have blood in your intestinal tract. So obviously if there's blood flowing in areas that it shouldn't be, then you could easily die. And so Ebola, this virus can infect numbers of tissues and different epithelial cells, but it does the most damage when it infects these macrophages, these immune cells, because then those lead to this really robust inflammatory response. So that can then cause the inflammatory response to kind of like that whole too much of a good thing. The inflammatory response can be activated and overstimulated, and that can then result in death. And so that's the underlying cause of the bleeding. But what you guys need to know is that Ebola is a virus, it's contagious, and that it causes this hemorrhaging through this immune response. Okay. So again, these pattern recognition receptors, they are receptors on the cell. So here we have a host cell, and so we have these pattern recognition receptors. The ones that, remember, are extracellular are the TLRs, toll-like receptors. Intracellular ones are nod-like receptors. There are some C-type lectin receptors. So there's three types essentially that you might see in figures, and they're TLRs, NLRs, and CLRs. But what you should know is that these receptors are expressed on the host cells. They also can be expressed by these macrophages as well, but they recognize those patterns that are on the microbes. So in order for a pathogen to actually make or cause the infection, it has to go through a specific mechanism and actually, subvert all of these different checkpoints. So first, the pathogen has to gain access to the body and find somewhere to colonize. So by gaining access, it's got to adhere or attach and then colonize. And so we'll go through these one by one. So we're going to start with this one. So this infection is often initiated by adherence. There's got to be some sort of adherence mechanism. So we talked about Peelle, they're little hairs on bacteria that are involved in adherence. The flagella are also involved in adherence. There's capsules. There's other cell surface adherence. So bacteria, they have a lot of things on their surface that can help them adhere. So here are pictures showing you flagella. These are Peelle and Fimbria, kind of similar. Peelle are just these longer ones. The Fimbria are the smaller, thinner hairs. You have capsules around bacteria. You can clearly see this is the bacteria and this is the capsule that it's made around it, so it's sticky. So the point is that you don't need to remember all of this, but what you should know is that bacteria have different types of adherence molecules, and those attach to a host receptor. There's certain places where they can attach, and they cause diseases eventually if they are successful in the rest of the process. But adherence also is not just mediated by these structures on the outside of bacteria. Remember we talked about biofilms, and biofilms actually play a big role in bacterial adherence. So they help enable that persistent adherence and resistance to the host trying to kill the bacteria. So you guys remember what biofilms are? They're essentially collections of bacteria that stick to each other, and so this is a mechanism for them to evade the host immune response and also for adherence. It's a really good mechanism for bacterial adherence because they form that extracellular matrix, remember, and so that then attracts other bacteria to come in and stick. So it's a good mechanism for them to survive. So now the pathogen has adhered and it's gained access to the body. It's found a niche and it has to colonize. So the next step is that it's got to acquire the necessary nutrients for it to be able to survive. And then once it does so, it's got to multiply and persist. So colonizing is one thing, but persistence is another thing. And those are something that's really important in the research field to understand. In vivo means in the body or in the model organism. In vitro, on the other hand, means in a dish or in the laboratory setting. So just a heads up there. So it's got to acquire the necessary nutrients and then be able to multiply and persist for it to become an actual infection. Okay, so they'll grow locally, or they could spread throughout the body, which is worst case scenario. But the availability of the appropriate nutrients is what's really important here. And so I had more on this, but I didn't want it to get too tricky, so I'll just leave it there, and we can move on to the next step for the pathogen, which is now that it's multiplying and it's persisting in that area. Now your immune system is going to start, at least the innate immune system is going to start, throwing cytokines into the mix and trying to signal that something is going on. There's alarm signals going on. So now the pathogen has to be able to avoid, subvert, or circumvent the innate host defense. So remember, the innate immune response is the first line of defense. And so there are certain ways that pathogens can avoid the immune response. For instance, we talked about phagocytosis. We have these macrophages that are able to phagocytose the bacteria. Well, we talked about capsules and biofilms. Those can help protect against phagocytosis. So the capsule of Streptococcus pneumonia, actually what causes strep throat, and pneumonia, protects the cell from being destroyed from phagocytes. So that capsule not only helps in adherence, but it also helps it from being destroyed by phagocytes. The same thing can be found in biofilms, because if the biofilm is in your body, then the immune cells can try to attack it, but it's not going to be able to attack whatever is underneath there. There's layers and layers of bacteria. So there are a lot of ways for bacteria to avoid the immune system. Sometimes they will actually be able to get internalized into an immune cell and just kind of not be seen. And it's called like a Trojan horse mechanism, where eventually the immune cells take it to other locations in the body and it hides out in the immune cell. There's this type 3 secretion system of bacteria where it's like having a molecular syringe, and the bacteria is injecting proteins into the host cell. So it's a mechanism of delivering certain proteins from the bacteria into that host cell. And so these are actually, it appears that this is an evolutionary mechanism that where the flagella were re-engineered in order to encode these proteins that act like these molecular syringes. You don't need to know that they're located on pathogenicity islands, but a lot of different infections cause this. So the way that this works is they're blocking phagocytosis. So they can stop phagocytosis by injecting the phagocyte, the host immune cell, with these proteins, which we're calling effectors. So here there's Yersinia pestis. This is a species. And so it basically has this molecular syringe. And this is the phagocytic cell. This is, say, the macrophage. And so it injects through the syringe these YOP, H and J, these effectors, these proteins, and so this then will block certain signaling pathways for the macrophage, And usually these are going to involve cytokine production, cell survival and activation, but if they're blocked, then you don't get that. So it's essentially disrupting the mechanism of phagocytosis and the signaling pathways that eventually will cause the rest of the immune cells to come and try to kill this pathogen. So this is one of the ways that you can block phagocytosis. So remember, the innate immune response is the first line of defense. So we're talking about macrophages, those neutrophils, all of those other phils that we talked about. But then what happens after the innate immune response is triggered is there are, remember, antigen-presenting cells like macrophages and dendritic cells that present those antigens to the adaptive immune response. So the next step is that the pathogen has to evade the acquired, which is the adaptive immune response. Because remember, this one is acquired through time, over time. One of the ways that pathogens do this is by hiding inside of the host cells. So a lot of times, so remember, the adaptive immune response includes T cells and B cells. B cells make antibodies. Antibodies act within extracellular spaces, but some pathogens can stay inside of the cell after it's ingested. So even if a phagosome or a macrophage or a phagocytoses the bacteria and it wants to present it to something, to the adaptive immune response, some of those aspects of the adaptive immune response only work on things in the extracellular space. So some pathogens hide out. And so one of those really key examples is listeriosis. It's this disease caused by Listeria monocytogenes. This could lead to bacteremia and meningitis. Bacteremia is essentially sepsis. You have bacteria all throughout the bloodstream, and meningitis is infection and swelling of the brain. This is found in soil and in water. It can grow at colder temperatures. Usually it's killed by cooking and pasteurizing foods. It can be transmitted by foods. So this is an important infection in really vulnerable, immunocompromised groups. But this one, what you need to know is that it's an intracellular pathogen, which means that it can be taken up by a phagocyte, and that can then allow it to grow and proliferate inside while it hides out. So this should say some pathogens hide out inside host cells. But essentially what's happening is here, remember we talked about this phagocytosis process. You have a bacteria, it gets engulfed and ingested by the phagosome or a phagocytic cell. And so it turns into a phagosome, and then you have the lysosome with these degradative enzymes that fuses with the phagosome to make a phagolysosome. And then that then destroys the microbe and releases it. Some pathogens can stop this phagolysosome from maturing, and it can then escape before it fuses with a lysosome. So it can replicate and do its thing in here and then escape. So that's what Listeria monocytogenes does. Okay, so once this pathogen has evaded the innate and the adaptive immune response, then it starts to cause this tissue damage or disease. And so this disease aspect is usually promoted by what we call virulence factors. So virulent means something that is disease causing. So there are enzymes and toxins and things that the microbe can produce that are virulence factors, and so these are what are going to promote this infection and host cell damage. So enzymes are anything that ends in ACE. Toxins, there's exotoxins and endotoxins. And so there are many different types of virulence factors that are encoded by genes in the genome of the bacteria that can kill or inhibit the host cell and cause that type of damage. So endotoxins are lipopolysaccharides, LPS, that's found in the outer membrane of gram-negative bacteria. Now we're kind of going back and linking some of these topics to the gram-negative, gram-positive cell walls of bacteria. So these endotoxins are released as the bacteria start to die, but they also serve as a MAMP, which can bind to PRRs on leukocytes. So that causes cytokine to release, and that could eventually cause this overstimulation of cytokines that triggers the fever, shock, and death. So that's that cytokine storm. Exotoxins are toxic proteins that are released from the pathogen as it grows to cause that host tissue damage and death. Essentially, they poison the cell. There's different mechanisms by which they act, and they essentially are causing damage to the host by destroying the cell or disrupting its metabolism, but essentially they are directly poisoning the cell. So how do these microbial exotoxins do this? Their mechanism of action? Well, they can interfere with really any part of the host cell. It could be, they can disrupt the plasma membrane, the cytoskeleton, protein synthesis, the cell cycle, signal transduction pathways, cell-to-cell adherence, vesicle traffic, exocytosis. There's so many different ways. So you just need to kind of be familiar with a list of them. Okay. I will post worksheet 7 today, and it will be due on Monday, and Monday we will review it. Any questions? All right, if you have not picked up your exam, I have it here. Have a good weekend.